Let's unpack this. We're
Yeah.
diving into the world's greatest, most unkillable survivalists. Seriously, forget the usual suspects, tardigrades, crocodiles, that weird Tupperware in the back of the fridge.
Right.
I'm talking about plant seeds. These are nature's ultimate kind of tiny architects of endurance.
Yeah.
And the stuff we found in our reading, well, it's going to change how you look at, like, the dust on your windowsill.
It genuinely does. It completely redefines what we even mean by resilience. You look at a seed, you think fragile, right? Maybe it doesn't last long.
Like, okay.
But physiologically, genetically, they are these perfectly engineered time capsules designed to wait out or sometimes even require like planetary scale catastrophes. They're the ultimate biological pause button, basically.
Okay, so to set the stage, we've got 3 facts to just throw at you straight away.
Yeah.
First, this plant, right, revived after 31,800 years in Siberian ice. Just think about that time frame for a sec. That seed was like chilling during the Pleistocene. It's older than the pyramids, older than basically any major landmark you can think of.
It's unbelievable. A tangible connection to deep time, like you said. But it also shows you just how much evolutionary investment goes into this waiting strategy.
Right.
And then you pivot to just the sheer absurdity of scale. We found documentation of a seed, 25 kilograms. The coco de mer.
25 kilos.
Yeah, that is like the botanical equivalent of lugging around a small cinder block, but it's a seed.
I'm trying to picture what disperses that, like
Yeah.
a small forklift maybe?
Huh. Probably something slow and steady, and then...
Your favorite? Oh, yeah. My personal favorite bit of botanical mischief, the fruit that literally builds up pressure inside itself until it explodes its seeds out, like a projectile.
Nature's own mini cannon.
Exactly. Using pure hydraulic pressure. Launches its little future offspring like 6 meters away. It's wild.
So, our reading details about, 40 incredible examples showing how life survives all this crazy stuff, drought, insane fire, deep ice, super salty conditions, often packed into something smaller than a grain of rice.
Incredible.
Our mission tonight, basically take a chill late-night look at these botanical oddities, uncover the specific physiological tricks, the morphological hacks these tiny architects use to just stay alive against all odds.
So yeah, settle in. We're doing a deep dive into the things that make you stop and go, wait, what the actual heck? Okay, let's start where the timeline just gets completely warped. Life under the ice. We got to explore the engineering genius that lets these seeds basically cheat physics, right? Survive millennia frozen solid in permafrost.
This is the territory of true cryogenic survival, and the undisputed champion, really, is the quote-unquote undead flower, Selene stenophylla.
Okay, the undead flower. Love it. And the discovery itself is just wild. Finding viable plant tissue in a fossilized squirrel burrow near the Colima River back in 2012. The shock isn't just that it's old. I mean, 31,800 years, that's the oldest verified plant regeneration ever. The real shock is finding viable life from the Pleistocene, basically just waiting to wake up.
And the ability to survive that long, it comes down to preventing cellular damage, mainly through this really high-tech mechanism called vitrification.
Vitrification, okay.
So most cells die when they freeze because the water inside forms these sharp hexagonal ice crystals, you can imagine, right?
Yeah.
Tiny shards of glass just shredding the cell membrane, the organelles, instant death.
Nasty. So the plant has to stop the water from crystallizing at all.
Precisely. Silene cheats. It rapidly dehydrates itself and pumps itself full of cryoprotectants. So at temperatures between, say, -20 and -60 Celsius, the water inside doesn't crystallize.
It doesn't.
No, it super cools, gets really thick really fast, and turns into this amorphous glass-like solid. It's not just frozen, it's chemically transformed into biological glass, stops all metabolism but without the physical damage.
Whoa. So it's like putting the cell into this perfect glassy stasis, pausing time without actually breaking anything important.
That's a great way to put it. And that stasis is fiercely guarded by some specialized chemistry. It's augmented by molecular protection, mostly these things called dehydrin proteins, part of the LEA family.
Dehydrins, okay.
And the amount is staggering. They can make up over 15% of the seed's dry mass. That's a huge chunk of resources dedicated just to this.
15%. What are these dehydrin proteins actually doing to earn that much of the budget?
Well, scientists call them molecular chaperones, but honestly, that almost undersells it. They're hydrophilic polymers, right? They bind directly onto the cell membranes, onto vital enzymes.
Hmm.
So when the cell dehydrates and vitrifies, turns glassy, these proteins kind of coat and stabilize all the critical machinery inside. They stop proteins from clumping together, stop the whole structure from collapsing. They're like the internal scaffolding that keeps everything perfect for, well, millennia of deep freeze.
That's structural integrity.
Mm.
Okay, that hasn't covered the DNA too, right? 30,000 years. Even low-level background radiation, oxidative stress, the genetic code must take a beating.
Oh, absolutely. It definitely suffers damage. But the seed isn't banking on, like, magical perfection. It's banking on active, systematic maintenance. Our reading points out that as soon as it rehydrates, the embryo kicks in these really robust DNA repair pathways, specifically base excision repair.
Wow.
This molecular machinery just goes through, finds the oxidative lesions, the UV damage accumulated over all that time, and snips them out, fixes them. It's like biological quality control, making sure the blueprint is sound before it commits all that energy to actually growing.
The fact that a seed, 1.2 millimeters across, is carrying its own repair shop, the architectural scaffolding, and the antifreeze chemistry for 31,000 years. That's just mind-bending.
And Silene isn't alone in this deep freeze warfare. There's the lemmings legacy, Lupinus arcticus, the Arctic lupine.
Ah, the lupine.
Mm-hmm.
Slightly less ancient, right? Only, quote-unquote, only 10,000 to 13,000 years old.
Relatively young.
Yeah. But equally stunning. Found in Alaskan lemming burrows back in the 50s. And that find that immediately forced scientists to, completely rewrite the book on how long seeds could stay dormant.
It really did. Lupinus arcticus uses similar cryoprotection chemistry. It focuses on these raffinose family oligosaccharides, RFOs.
RFOs, got it.
These are super effective sugars. They don't just lower the freezing point, they actually help promote that glass formation in the cytoplasm, help it reach that stable vitrified state. And they also use antifreeze glycoproteins. These kind of latch onto any tiny ice crystals that might try to form and stop them from growing.
Okay, so the chemical protection is sorted, but the lupinus also has this legendary physical protection, doesn't it? Like it's wearing medieval armor or something.
It's basically an armored vault. Yeah, its physical dormancy comes from this multi-layered, totally impermeable seed coat made of these densely packed cells called macrosclerids.
Macrosclerids, like tiny bricks.
Kind of like microscopic, tightly woven rebar rich in lignin. This physical barrier keeps the inside humidity critically low, below 5% for millennia. That stops destructive enzymes, stops mold, stops fungus, just keeps it safe and dry.
Okay, safe and dry, frozen in time. But now that armor is the biggest problem for its own future, right? How does it ever break that seal and actually germinate?
It needs scarification. The seed coat has to be physically broken or worn down.
Scarification, like getting scratched up.
Exactly. In the subarctic where it was found, this usually happens over thousands of years of just constant freezing and thawing. The ground expands, contracts, expands, contracts. Eventually, it cracks the armor. Or maybe if an animal eats it, the grinding in the digestive tract could do it. It literally takes an act of, geological scale violence just to wake this seed up.
That whole idea that life needs this sustained environmental catastrophe just to start its next phase.
Yeah.
That's an incredible evolutionary trade-off. It's designed to wait out the calm and just embrace the chaos.
It is. It really is. The resilience you see in these cryo-survivors, it sets a seriously high bar for the rest of our deep dive tonight.
Definitely. Okay, before we move on from the deep cold and pivot to fire, I got to stop and just ask you, the listener, what biological blueprint blows your mind more here? The sheer time scale, that 31,000-year wait, or the, complex chemistry of turning yourself into glass? What do you think? Seriously, comment below. We're a small channel, and we genuinely love to hear from you. Love to have you on board. So please, like and subscribe if you're enjoying this. If these tiny seeds can survive the last ice age, you can probably hit a button. We really appreciate it. All right, so from the deep freeze, let's pivot hard to the other extreme. Fire, we're moving into seeds that don't just, tolerate fire. They're fundamentally pyrophytic. They need it. It's their wake-up call. Heat is the key, the perfectly timed key to unlocking their future.
Yeah, it's truly counterintuitive, isn't it? Humans, we universally see fire as just destructive. But for these fire-adapted ecosystems, it's programmed renewal. The classic example, jack pine, Pinus banksiana.
Ah, jack pine. Textbook case of Serotyny architecture. You see those cones, right? Sealed tight. Sometimes they hang onto the tree for decades, holding the seeds hostage.
And that seal is the key. It's this ligno-resinous matrix. Basically, a super tough glue made of abiotic acids. It cements the cone scales together. They only pop open when the temperature right on the surface hits the resin's melting point, which is very precisely calibrated, actually, between
Wow.
49 and 63 degrees Celsius.
49 to 63 C. That seems kind of low. Like you could melt that with a hot hair dryer, maybe. But forest fires get way hotter, hundreds of degrees. How do the seeds inside survive long enough for the cone to melt open and drop them onto, a good spot?
That's the survival paradox right there.
Mm.
While the surface temp of the fire is absolutely lethal, the cones themselves, they can actually withstand brief exposures up to 500 degrees Celsius.
500.
Because that thick, dense resin acts as a surprisingly effective insulator. The material heats up slowly, protecting the little winged seeds the samaras tucked inside. Those seeds can stay viable in there for over 25 years, just waiting for that perfect hot moment. The fire becomes the catalyst for this immediate mass dispersal onto ground that's been perfectly prepped, cleared of all competition, super rich in nutrient ash.
That is some next-level patience and planning. Okay, let's talk serious heat, though. If Jack Pine is tough, the Australian Banksia is just, well, it's insane, surviving those infernos in the Australian heathlands.
Oh yeah, the Banksia species are the armor-plated vaults of the plant world. They're woody structures that hold the seeds. They're called follicles. And they're reinforced with dense sclerenchymatous tissues, plus they're fortified with silica phytoliths.
Phytoliths. Those are like microscopic bits of rigid silica, right? Like tiny glass fibers woven right into the structure.
Exactly like that. The silica acts as a refractory barrier. It resists heat incredibly well. Our reading notes these follicles can withstand inferno temperatures up to 800 degrees Celsius
800 degrees.
for up to 2 minutes without compromising the embryo inside. That's heat resistance engineered right down to the cellular level. It's phenomenal.
Okay, so the heat melts the glue holding the follicle shut, but the structure itself survives the blast furnace. But we also know the fire isn't just about clearing the path, is it? It's also sending a specific chemical start signal.
Ah, yes. This is maybe the most brilliant part. The environment after a fire produces specific volatile compounds in the smoke. The key molecule is this butanolide called keratin. Kerikins actually mimic naturally occurring hormones inside the seed called strigolactones.
So the seed's dormant, basically asleep. Then it smells like the chemical signature of burnt wood, burnt cellulose. And that molecule fits perfectly into a receptor inside, like a key, telling it, go, now's the time.
Precisely. Kerikins are recognized by these specific KAI2 receptors. That molecular signal immediately breaks deep physiological dormancy. It often boosts germination rates way up, like 90% or more, right after a blaze. So the fire gives the heat to release the seeds and the chemical trigger that guarantees they all grow fast, synchronized into that fresh, nutrient-rich ground. It's not just surviving fire, it's engineering a system that absolutely requires fire for success.
Wow, an ecological symphony of destruction and creation,
Yeah.
basically.
So just to round this section out, we should probably give a quick shout out to a couple of other hardy examples that show this fire codependence.
Definitely. There's Hespercyparis macrocarpa, the iconic Monterey cypress.
Yeah.
Another specialist using those serotinous cones for fire release, but in its very specific coastal California home.
Right.
And then you have the ultimate post-fire successor way up north, Betula piperifera, the paper birch.
Ah, the paper birch. Beautiful tree. Thrives in the taiga, right? It uses fire to clear the way, but then it has to deal with an insane cold afterward, down to minus 50 C. It's the phoenix of the freezing north, absolutely. Fire clears out the competing trees, gives it the light it needs, and then its rapid growth lets it just dominate those ash beds before anything slower can get established. It really proves that resilience often means being adapted to both extremes of temperature once the competition's been wiped out by the heat.
Okay, now let's tackle maybe the most relentless stressor on the whole planet.
Yeah.
Just absolute baking, desiccating drought, and often high salinity, too. We're talking desert dominators now. The ultimate thirst quenchers, using bioengineering to survive places that should be, totally hostile to life.
Drought really is the great equalizer, isn't it? And these seeds, they've evolved two main strategies, kind of divergent. Either extreme patience hydrodormancy or this hyper-efficient, really localized water harvesting.
Let's start with patience, the waiting game, represented by Astragalus lentiginosus, the freckled milk veg.
Right. This species shows incredible longevity. Seeds can wait in the soil in the seed bank for up to 50 years.
50 years.
But its real trick is this strict, hydro time threshold it sets for itself. It won't even think about activating its metabolism unless it detects a really significant sustained rainfall. Typically, that means over 10 millimeters of rain.
So if it just gets a light sprinkle, like a millimeter or two, it just ignores it.
Completely.
Refuses to waste its shot on a false start.
That's exactly the calculation. Germinating after just a little bit of rain, a fleeting pulse, that's a guaranteed death sentence in the desert. The topsoil dries out way too fast, so the seed stays deep in physical and chemical dormancy until that specific, reliable environmental cue arrives. The cue that guarantees there's enough moisture for the root, the radicle, to emerge and get established for at least a few weeks. It's the ultimate calculated gamble on the water cycle.
Wow. Okay, now contrast that extreme patience with the super sponge strategy used by Plantago ovata, the desert plant most people know as psyllium, right, for the husks.
That's the one. Plantago ovata has mastered mixospermy. The seed itself is tiny, like 2 millimeters long, but the second it touches moisture, it oozes this mucilage coat. It's a hydrophilic polysaccharide layer. Incredibly complex stuff.
How super is this sponge we're talking about?
It expands up to 100 times its original volume.
100 times?
Yeah. And that gel coat does two amazing engineering jobs. First, it anchors the seed really firmly, stops it blowing away or washing away. Second, it acts as this ultra-efficient water harvester. It literally scavenges dew, any tiny bit of ambient humidity.
And this lets it deal with something that kills most plants. This idea of severely negative metric potential in the soil.
Exactly. And we should maybe clarify what that means. Metric potential, it describes how tightly the soil particles are holding onto water molecules, the capillary tension. When that potential drops to minus 1.5 megapascals, minus 1.5 NPA, that's the classic permanent wilting point for most plants that aren't adapted. It means the soil is clinging so fiercely to the water, the plant just physically cannot suck it out anymore.
But the Plantago seed cheats this whole system.
By creating its own personal, localized little gel bubble, the mucilage ensures that the critical first step, the root emergence, happens even when the surrounding soil has that extreme water tension. It's basically engineering a microhydrated oasis right around itself, using its own biochemistry. It effectively ignores the extreme drought just millimeters away in the rest of the soil.
That level of ingenuity, it's not just for wild plants, is it? These hard-won tricks are actually vital for our future. Our reading highlights some modern staple crops that have naturally figured out similar techniques.
Absolutely. First one is sorghum bicolor, sorghum. Its secret weapon is its C4 Krantz anatomy. Most plants just suck CO2 out of the air, that's C3 photosynthesis. But sorghum has a specialized leaf structure. It actively concentrates CO2 internally using two different kinds of cells, the bundle sheath cells and the mesophyll cells.
Like having a specialized biological turbocharger for CO2.
That's a perfect analogy, yeah. This structural tweak lets it achieve, quadruple the photosynthetic efficiency in high heat and with minimal water loss through its pores. Plus, it has the stay green QTL quantitative trait loci. These are genes that basically delay the breakdown of chlorophyll when stressed. Helps it survive long droughts, keep producing grain when other crops would just wither and die.
Okay, then you've got the halophytic Highlander, great name. Chenopodium quinoa. Quinoa handles high-altitude cold and extreme salt.
Quinoa is just a powerhouse of resilience. Thrives in the Andes, 4,000 meters up, endures soil salinity up to 40 DSM that's incredibly salty and frost down to minus 8 Celsius. Its secret weapon is accumulating this osmolite compound called glycine betaine.
Glycine betaine, tell us about that because that sounds key for salt tolerance.
It's what's called a compatible solute. So when the plant takes up lots of salt sodium, chloride ions, those ions mess up the osmotic balance inside the cells. They basically try to pull water out of the cell, and they interfere with that enzyme's work. Glycine betaine builds up inside, sometimes up to 200 millimolar concentration, and it acts as an internal stabilizer. It balances the cell's internal water pressure against that extreme external salt pressure. It stabilizes critical proteins and membranes, but crucially, it does this without messing up normal cellular chemistry. It's compatible.
So it's basically acting like a natural internal antifreeze and anti-salt chemical, fighting heat, cold, and ionic stress all at once. A true bioengineer.
Absolutely. And finally, just quickly, we can't forget the extreme dispersal methods that desert life drives. You contrast that patient waiting game with these really aggressive, sometimes chaotic strategies needed for rapid colonization.
Like Salsula tragus, the Russian thistle, which everyone knows is the tumbleweed, right?
Exactly, that classic invader. The whole parent plant just detaches after it matures, and the resulting rolling ball of well, chaos, just scatters seeds everywhere as it tumbles. Up to 250,000 seeds per plant. It's high volume, chaos-driven distribution across the plains.
250,000 seeds.
Hmm.
And for pure speed, you mentioned Euphorbia heterophylla. Doesn't wait for wind or rolling. Its pods just explode.
Literally explode. They build up enormous hydrostatic pressure inside, and then bam, they launch their seeds at 10 meters per second. That rapid ballistic dispersal, it's called bolocry, ensures those seeds hit the ground running, basically. Grab those fleeting bits of moisture before anything else can. In the desert, speed is life sometimes.
Okay, we have covered the really hard science of survival, right? Ice, fire, drought. Now we get to my absolute favorite part of our reading, the wonderfully strange, the highly specialized, sometimes frankly hilarious adaptations. This is the weird shot that works section.
Yeah, we're definitely moving from pure hardiness into the realm of evolutionary absurdity now. And where better to start than with nature's canon? The spectacular high-pressure dispersal of Ecbalium elaterium, the squirting cucumber.
Squirting cucumber. It sounds like a biological water balloon fight gone very, very wrong. How does it build up that much pressure?
So the fruit, it's a small thing called a pepo, and it builds up this astonishing internal hydrostatic pressure, turgor pressure, up to 1 megapascal.
1 megapascal. How much is that like in real terms?
Well, standard atmospheric pressure is about 0.1 MPa. A car tire, maybe 0.2, 0.25 MPa. This little fruit generates nearly 10 times atmospheric pressure inside itself.
That is a serious amount of pressure for plant cells to contain. What triggers the squirt?
When the pressure peaks, the fruit basically inverts its entire internal structure right at the stem end, pops off, and it happens in about 40 milliseconds. Instantaneous failure. That jets out the seeds, which are coated in mucilage about 6 meters away, traveling at roughly 10 meters per second. And this whole mechanism, it evolved specifically as defense and dispersal, especially good against slow movers like snails in its native Mediterranean habitat.
It built a finely tuned hydraulic pump just to blast its enemies and spread its seed. Evolution is delightfully aggressive sometimes.
It can also be kind of sinister. We have to mention Araugia cercifera, the cruel moth trap.
Ooh, the name alone promises some drama there.
Well, its follicles invert to release the seeds, and they have these silky little parachutes, coma tufts for wind dispersal. Pretty standard, but the flowers themselves, they're structurally designed to trap moths and other big flying insects. The insects get stuck trying to feed and often just die there, hence the nickname, the cruel plant. It's actually quite a beautiful plant, but yeah, it's got this dark side. And it can be pretty invasive, too.
Okay, moving from the tiny and sinister to the just, outright absurdly large, we revisit the behemoth, the mega endosperm of Ladoia maldivica, the coco de mer.
Right. The sheer physical presence. That double coconut. 25 kilograms, 40, 50 centimeters across. It seems completely incomprehensible, right? Until you understand the strategy behind it.
Yeah, why is such a massive seed? A normal palm seed is like tiny in comparison.
It's the ultimate investment in provisioning. It needs that huge mega endosperm because it requires 2 years of sustained internal energy reserves just to germinate and get the first seedling established.
2 years just to sprout.
Basically, yeah. It doesn't need rapid dispersal or small size because it grows in this very protected, stable environment, the Seychelles Islands. This massive energy deposit ensures the seedling can survive that whole initial establishment phase without needing much from the soil right away, which is critical in its kind of unstable, rat-dispersed island home.
That actually makes perfect sense. If you've got enough internal fuel for a 2-year head start, you don't need speed, you just need sheer volume, reliability.
Exactly.
Yeah.
Now, for maybe the most shocking dispersal mechanism we came across, the toucan palm, Astrocarium aculeatum. This one involves ichthyocrae, fish dispersal, specifically piranhas in the Amazon.
Wait. A plant that relies on piranha guts for successful reproduction? I need the details on this one. This sounds insane.
It's pretty wild. So the piranhas eat the fleshy outer part of the fruit, the drupe, that surrounds the hard seed. But crucially, that hard protective layer of the seed, it needs chemical scarification to break its dormancy and get it ready to germinate. Guess what provides the absolute perfect combination of physical abrasion and digestive acids?
The piranha's digestive tract. The predatory fish is actually the plant's essential pre-processing partner.
You got it. The journey through the gut prepares the seed perfectly for germination in the warm, like 26 degrees Celsius, waters of the Amazon floodplain forest, the Várzea. The seed is literally using a dangerous predator as its dedicated gardening assistant. It's a brilliant biochemical cheat.
That's just brilliant. Okay, finally, we should briefly mention the seeds that try not to be seen at all. Crypsis and camouflage, like in Lithops spp, the living stones, and Euphorbia obesa, the baseball plant.
Right. In super arid places like the Karoo Desert in southern Africa, survival often just means avoiding being eaten. So these plants evolved to look exactly like pebbles. That's crypsis. And they often use geocarpy, which means ripening their fruits either underground or really close to the ground, making sure the seeds are just never found by hungry herbivores.
Hiding in plain sight.
Exactly. And lithops seeds, they only open their capsules when it rains hygrocastic dispersal. The moisture makes the capsule pop open, meaning they only release their future generations when germination is pretty much guaranteed. Masters of disguise and masters of hydraulic timing.
Okay, we have established, I think, beyond any reasonable doubt, that seeds are basically the toughest things on this planet, using glass, fire, fish guts, pure pressure, you name it, just to survive. But the big question always remains, so what? What does all this mean for us? Why do these, like, wild botanical oddities actually matter to humanity? From modern farming to maybe even space exploration.
They matter enormously because they are these time-tested blueprints of resilience. They show us highly optimized solutions to some of the biggest challenges humanity is facing right now. Water scarcity, soil salinity, just surviving in extreme environments.
Let's start with coastal guardians.
Yeah.
You mentioned Rhizophora mangle, the red mangrove, salinity tolerance. That's incredibly hard to engineer into crops, right?
Yeah.
Yet these plants literally live bathing in 40 parts per thousand saltwater.
Right. And mangroves kind of bypass the whole deadly soil salinity problem by using vivipary.
Vivipary, that means the seed germinates while it's still attached to the parent tree.
Exactly. It develops into this large, pencil-shaped thing called a propagule. It's actually a specialized embryonic root, the hypocotyl, up to 30 centimeters long before it even drops off the tree.
So it's basically an autonomous, fully established little seedling before it even hits the water.
That's the genius of it. This propagule can then float around for up to a year, staying viable in highly salty water. When it finally lodges in some coastal mud, it's already way past that super vulnerable, tiny seedling stage. This system doesn't just ensure dispersal, it's fundamental to stabilizing entire tropical coastlines against erosion, against storm surges. It's a massive ecological service built right into the seed's strategy.
And these natural solutions, they're now forming the bedrock for a lot of modern biotech, aren't they? We're actively learning from these extremes to try and shore up our own food supply.
Absolutely. The biotech angle is where this deep dive into resilience really starts to pay dividends. For instance, those drought tolerance genes we talked about, like the RFO synthesis pathways from the Arctic lupine or those mucilage traits from Plantogo ovata, they are critical targets right now.
Scientists are actively looking at using tools like CRISPR editing to insert or enhance these specific genetic pathways into staple crops, things like maize, wheat. So we're basically trying to borrow the super spongeability from that little desert plant and give it to corn.
That's precisely the goal. The FAO reports that targeting genes related to those LEA proteins, the molecular chaperones from the ancient Silene, or the genes for glycine betaine production, like in quinoa, doing that could significantly improve a crop's ability to regulate its own water balance and maintain yield, even in severely water-scarce regions. We're essentially taking millions of years of evolutionary R&D and trying to fast-track it into modern agriculture.
And look at cryopreservation, too. The whole idea of deep freezing our global food biodiversity in seed vaults that relies entirely on the lessons learned from these extreme cold survivors, doesn't it?
It absolutely does. The sophisticated protocols they use for long-term storage in places like the Svalbard Global Seed Vault, maintaining ultra-low moisture content using rapid cooling rates, they are directly inspired by that natural vitrification mechanism, the one discovered in that 31,800-year-old Silene seed. We're using deep history to try and safeguard our future against things like rapid climate change.
Okay, and here's where it gets really interesting for me. Space. How do these incredibly hardy seeds on Earth connect to the potential for, like, extraterrestrial agriculture? Farming on Mars.
Well, NASA, other space agencies, they've been doing experiments, including recently on the ISS in 2021. They study model plants, often Arabidopsis thaliana. It's a small, common mustard weed, grows fast, well-understood genetics. They focus heavily on its ability to develop things like flavonoid UV shields and its general hardiness against radiation.
Flavonoids, those are like internal sunscreens, right? Antioxidants, too. In space, that radiation shielding must be absolutely critical.
Crucial. The environment in orbit, or on the surface of Mars. You've got Vacuum, huge temperature swings, and intense cosmic and solar radiation. The genetic mechanisms that allow Arabidopsis to produce those robust flavonoid layers, and importantly, to maintain those DNA repair pathways we discussed earlier, they draw direct parallels to the adaptations we see in Earth's toughest seeds from extreme environments. Understanding this resilience is genuinely paving the way for eventual agriculture in orbital habitats, maybe even on other planetary surfaces someday. We're taking the world's quietest, toughest organisms and preparing them for a potential stellar career. So if we kind of synthesize this whole deep dive, I think the ultimate takeaway is just the staggering complexity hidden inside the smallest packages imaginable. Whether it's those systematic DNA repair systems working away in 30,000-year-old Siberian ice, or the precise chemical signals from smoke that basically demand instant germination, or those molecular stabilizers fighting off high salt. Resilience isn't just an accident. It is deeply, deeply built into the blueprint of life itself.
Absolutely. We've seen nature use amorphous glass, scorching fire, the incredibly acidic guts of piranhas, pure concentrated hydraulic pressure, just to make sure its future gets a shot. It's humbling, honestly, and kind of awesome. A reminder that the world's quietest organisms are often, by far, the most tenacious, the toughest.
And that, I think, leads to a final, maybe provocative thought for you to carry with you tonight. If life can be frozen solid for 31,800 years and then successfully revived, what other vast, dormant blueprints are waiting right now? Hidden beneath the earth, maybe in glaciers, maybe deep in the oceans, blueprints that hold keys to surviving the next planetary crisis, whatever that might be. We really need to be aggressively preserving these natural libraries, these genetic archives, because they almost certainly hold solutions to problems we haven't even learned how to ask for yet.